All right, we're back. Let's finish this off. I think that we can put everything into one longer lecture, although I might stop in the middle. We'll see what happens, okay? So we finally get to talk about the medical side of this. It takes a while to get to the idea that pH can change the charge on a drug, but it's hugely important because the uncharged form of the drug can pass across a membrane. And even if there's only a small amount of that uncharged part, uncharged form of the drug, it can still, the small part of it can cross the membrane and then change back, and then it's inside the cell. And so this is hugely important to how drugs work, how cells work. So the thing is, you do have some transporters that actively bring the drugs in or just open up big doors in the membrane and let the drug passively diffuse through the transporter. Usually we actually call it, if it's going through a transporter, we call that active transport. There also is a possibility for drugs that are lipophilic enough to pass through the membrane. If they are charged, it's very difficult, very energetically unstable for them to pass through a membrane passively. Small drugs and or small molecules and or nonpolar molecules can pass through the membrane. But of course, how can they pass through the blood to get to the membrane in the first place? We have to be going back and forth between different kinds of solubility for us to have a good drug that can get to the cell, get into the cell, and then get to the enzyme that it's going after. One of the other parts of this that I'd like to mention is that there are some cells that actively take in parts of the environment. They take in the extracellular parts by forming little vacuoles called endosomes. These endosomes actually have an important role to play that we're later going to talk about. But that is a particular kind of cell that is trying to bring in part of the outside of the cell into the cell. And it's part of the trafficking into the cell. I'm actually going to talk about this both at the end of this lecture and in the immunogenetics lecture as well, because endosomes have a role to play in both of those cases. The only interesting thing I'll mention about the endosomes right now, they change their pH. Most endosomes will acidify as they enter the cell. That will, of course, change the protonation of whatever molecule's in there. But just remember, we're mostly talking in this first place about how drugs can cross the membrane passively passing, diffusing through the membrane. Hard thing to do. So how this usually works, this actually explains how many drugs work. Acidic drugs will often be orally available. That means you can take them in through the stomach and then they can pass through into the body. And some of the drugs pass through better because they are acids that can actually accept protons and become uncharged. The idea behind this is an area of pharmacology called ion trapping. Now, remember that most drugs are too large to just get into the cell. If they're small, they might be able to sneak through. But most drugs are too large to pass through the membrane channels. They must diffuse through the lipid portion of the cell membrane. That means they must be non-ionized, at least in a minority fashion at some point. Non-ionized drugs are the ones that are lipid soluble and can pass through membranes. Ionized ones are the ones that are water soluble, and they are lipophobic, and they are lipid insoluble. Those two terms mean the same thing. Here's how understanding what pKa does for you helps you understand how an acidic drug can actually pass through the membrane if the acidic drug is taken orally. This shows you if you have an acidic drug that comes into the stomach. It starts off, let's say that you eat a drug that has an ionizable group. You know, in the saliva, in the mouth, and in the food most likely, it is probably at neutral pH, which means the pKa of that group is around 2 or 3. At neutral pH, that group will be deprotonated, and because it's acidic, it will be charged. And that will allow it to actually dissolve well in water. But when it gets to the stomach is a low pH environment. And so what you see happening to that deprotonated drug, it will then be protonated in the low pH environment, let's say you have a pH 1.4 gastric juice, and you have a weak acid, which is a pKa 4.4, kind of exactly like the acetate that we mentioned in the last little part of this. The weak acid will actually become protonated in the gastric juice. And so you can actually calculate exactly what the ratio would be with the Henderson-Hasselbalch equation. The HA will be in a 1 to 1,000 or a 1,000 to 1 ratio, 1 to 0.001, but that ends up being a 1,000 to 1 ratio, 1,000 times more protonated in the stomach. Of course, because it's acidic, when it's protonated, it becomes neutral, and it's able to actually pass through the lipid barrier into the plasma. This is greatly simplified, but it shows you how the pH could work. And once it gets into the plasma, all of a sudden, it's in a pH 7.4 environment. Then all of a sudden, the Henderson-Hasselbalch shifts around. If you calculate out what that's going to be, that ends up being a 1,000 to 1 ratio. On the other side of the ratio, you end up with a 1,001 of the deprotonated version of that. And lo and behold, the drug becomes deprotonated, it becomes soluble, and it cannot cross the lipid barrier again. So it is trapped in the plasma. It can't go back. And so you see how the different pHs of the different compartments and the fact that the protonated version is lipophilic allows drugs to be orally available. The ionized form becomes trapped on the high pH side. This is actually showing how you can use the Henderson-Hasselbalch equation to actually calculate what the ratios of the drug is going to be. You can actually calculate the ratio of A to HA. If you rearrange the equation, it becomes 10 raised to the pH minus the pKa, right? So in the plasma, that ratio is 10 to the third because the pH is 3 units higher than the pKa. And the ratio ends up being 1,000. And the top part of the ratio, which is the majority part, ends up being the A minus deprotonated form. In gastric juice, the situation is the exact opposite. The pH is 3 units below, you have a lot more protons, you have a lot more of the protonated form. And this is what allows drugs to actually be trapped in the plasma. You see, this would be very good for an acidic drug. So remember that acids are going to work this way. Drugs that are bases are going to work in the opposite way. Now, realize that there's a lot more things going on, so you can't just apply ion trapping to all of these drugs and understand completely what's going on, but if understood properly, it should be at least a part of what's going on for these stronger acid drugs that have pKa's in the range of 2 to 5. So that would include penicillin, salicylic acid, acetylsalicylic acid. So aspirin, I believe this explains why aspirin passes so easily into the body from the stomach. Aspirin is an acid. A lot of acids will do that. Those are in the orange box on the left. In the green box on the right, we have bases that actually have pKa's in a range close to physiological. And I want you to notice that you have several malaria drugs there. And I'm going to talk about how those drugs relate to pKa. Those are going to be different because when they are protonated, they are charged. They are unprotonated, deprotonated at higher pHs and protonated at lower pHs. So they have the opposite relationship of charge to pH because they are bases. They have the same ratio. The protonated form is still on the bottom of the Henderson-Hasselbalch ratio, by the way. But notice that quinidine, quinine, and quinine, they actually have 2 weak bases on them. And so we'll have to talk about how that works. How do they ever get across a membrane? Well, actually, we can take the information about them and we can figure out not only how they get across, but where in the membrane will they be if we do things like create artificial membranes for them. That's where this becomes really useful. So realize that there is a different behavior for drugs for acids and for bases. Remember the basic thing, or the fundamental thing going on here. For acids, A minus is charged, HA is uncharged. For bases, the A, the unprotonated version of it, sometimes it's shown as B, is uncharged, and the protonated version is charged. And that's what the cell cares about. So most of the nitrogen-containing groups physiologically are basic. That means they will accept a proton and become charged. And so amines are this way. Imidazole, which is the histidine side chain, is this way. And guanidine groups are this way. This will change which charge is on the molecule and how it will partition. So let's talk about those malaria drugs. The malaria drugs are going to be basic, and they actually have two nitrogens that can accept electrons, that have pKa's close to physiological. This is a complex figure that I want to explain, but it actually shows specific things about malaria. And so this is something that you may need to know in more detail later. I'm mostly interested in what this shows about the barriers a malaria drug has to pass through. Even if it's delivered intravenously, if it's in the blood, it still has to pass through many layers to get through the red blood cell membrane to where the malaria parasite is. Depending on where its target is, it may have as many as 4 membranes to pass through. They are numbered 1 through 4 this way. So the drug can get in, again, through the red blood cell membrane, and the first barrier it has to pass is the plasma membrane of the red blood cell. And so that can be either, there are some transporters that will transport in the drugs, but often there are also lipid diffusion. Sometimes that's not an option, the drug doesn't use it. Lipid diffusion can account for that. Then it has to get into the malaria parasite, and there are actually 2 different membranes on the outside of the parasite in the form inside the red blood cell. And once it gets past those 2 membranes, membranes number 2 and membrane number 3, it will actually possibly have a 4th membrane. It has to get into one of the organelles that sometimes are surrounded inside the parasite. It will have organelles that also have membranes. That's a lot of membranes. We're going to be mostly focusing on the very first step, where we have lipid diffusion of the drug across the membrane into the red blood cell. But it's worth pointing out, this is just one of the barriers that the drugs must overcome. So we don't even know necessarily all the barriers that the drugs must overcome. We just know that they work, and then we try to figure out how they work. So I want to mention these membranes if you're interested in malaria physiology and how that all works. You can identify the places the drug must cross. But for our purposes here, I'm going to focus on number 1, lipid diffusion across the red blood cell membrane. All the rest of the stuff just shows you this is a lot more complex than this one step can show you. So one of the things they've noticed is that the antimalarials that work and are approved, they tend to look the same chemically. They tend to have similar chemical structures, and they just look like they look the same. Part of this is that many of them operate on the same enzymes inside the parasite, or they operate using the same chemical means. So this means that there's something similar about them chemically. And in fact, this is even put on a graph right here, where you have a graph where you have the x-axis is polar surface area. And for the drug, this will just be the area of it that is carbon-hydrogen bonds and not able to hydrogen bond with the solvent. The higher the polar surface area, the more soluble it will be, but the harder it will be to get across the membrane. And you notice how we have all of these different drugs plotted out on this graph, and they tend to be 100th on this polar surface area scale or below. We have this sort of circle drawn around where most of the drugs are. And you see that most of the points are below 100 on the x-axis. That means they have low polar surface areas. So it makes sense that lots of these might get into the red blood cell by the passive diffusion method, because a low polar surface area goes with lipophilicity. There's also other ways to measure lipophilicity, and so there's some of these things are pharmacologically, they're called P98 values. And so there's one version of those that was put on the y-axis here. And you see that the higher the value, the higher the lipophilicity as measured, estimated by the value. And you see that these tend to be to have higher lipophilicities than some of the other molecules that don't quite work as well. I want you to be focusing, by the way, on the approved antimalarials, which are the green circles, and the blue triangles, which are compounds in the malaria pipeline. So they're probably going to be good drugs. And you notice that compounds in the pipeline tend to look like the approved drugs. They tend to look like they're chemically similar and similarly lipophilic and non-polar. The red squares are for comparison. They are antimalarial toxins, and these probably work using a different chemical mode. You'll notice that they tend to be outside the ellipse, and they tend to be on the extreme end, on this right end of the graph. Focus on the green dots, however, because the approved drugs shown include 3 drugs that we're going to look at in a second: chloroquine, quinine, and tafenoquine. I'm not quite good at pronouncing these because I am I'm a biochemist. I'm really not a pharmacist, but you can probably pronounce them better than I can. The other things to notice, let's see, actually one of those is a blue triangle. The tafenoquine is actually a blue triangle, but the major point is they are probably somewhere in that ellipse, that brown circle that's shown. That means that they have low surface areas, low polar surface areas, and high lipophilicity. So this is from the paper itself. They say, most existing antimalarials have physiochemical properties consistent with lipid diffusion. It looks like these get across the membrane passively. So based on this, the paper that I'm talking about is talking about an experimental strategy for getting these particular lipophilic drugs into the red blood cell in the first place. So it's a challenge for the drug to travel through the blood because it's not soluble in the blood. It's more soluble in membranes. So why don't we make an artificial membrane or a liposome? Another word for an artificial membrane is a liposome. A liposome is a vesicle. We talked about that at the beginning of the membranes lecture. Why don't we make an artificial version of a membrane that will be able to both carry the drug and then diffuse into the red blood cell membrane, perhaps. At the very least, get the drug into the membrane, and then we have a much greater chance of the drug actually getting through the red blood cell and to the malarial parasite. This shows how the liposome vesicles that this group made would work for hydrophilic molecules. So these would actually work for hydrophobic or hydrophilic molecules. For hydrophilic molecules, they'd work like this. If you actually form them at low pH, you see the yellow, if you form them at low pH, and then you put them into a 7.4 neutral pH buffer in the middle, the outside, of course, becomes neutral pH, but the inside, remember, the water has a hard time diffusing out, and those protons aren't going to be able to diffuse through the membrane, and so the inside will remain yellow. It will remain low in pH. And so when you actually load it up with drugs, if you're able to get the drug, maybe you increase the concentration of the drug so high that this charged, protonated, very hydrophilic drug, you can push it into the liposome by loading it up, and then actually the way they push it in, they show it, it's by ultracentrifugation. So once you do that, you have a high concentration of the drug in part C on the right, a high concentration of the hydrophilic drug in the water fraction inside the liposome vesicle. And then they can deliver, the other parts are, they're showing, then they can attach an antibody that will help it attach to the red blood cell, and then this would help to carry and deliver the hydrophilic molecule to the red blood cell. That's all well and good for hydrophilic molecules, but this includes a membrane. So couldn't the membrane dissolve hydrophobic molecules? And that's what they actually are looking into. So they're looking at using the liposome as a membrane that can deliver a little membrane bubble that can deliver the drug to a red blood cell, even hydrophobically, because the liposome has all this hydrophobic part to it. So they show that they can do a similar kind of loading where they do this, and they form the vesicles. On part D, they show forming the vesicles at low pH like they did before, but also mixing in the unionized forms of the drug. If you have an unionized form of the drug at that pH, what will happen is the drug will be everywhere. And so you'll end up with a low concentration of the drug inside the vesicle, but you'll see that you'll get a lot of the drugs. They're shown in red and orange. Orange is the singly protonated, singly charged form of the drug, and the red is the uncharged, unionized form of the drug. And so you see that actually both of those will dissolve in the membrane, and then when you take the membrane into a neutral solution, say the blood, it will maintain its pH 4.0 inside, and it will also be able to carry both the red neutral zero charge molecules and the orange singly charged molecules to the red blood cell, and the membranes will then merge with the liposome, and it will actually deliver it to the red blood cell. It's actually a pretty interesting strategy, and it builds on the way membranes work. What I like about this is it shows you how membranes work, because that's what we're after here. So if you look at this, there's one other thing you might notice about this figure. Look where the red and the orange are, especially on the right side of the figure in E and F, when you have the vesicle, the liposome, in a neutral solution. You have some of the red dissolved in it, but you have all of the orange on the inside. It's on the inner leaflet of the liposome. Why are they saying that? Well, I'm going to show you two reasons why they say that. One of the reasons why they say that, you can say it right away, that the pH inside is 4.0, so that has a lot of protons. It will protonate the drug. But the drug, why does the protonated drug stay on the inside of the membrane? And where does the protonated drug go once it gets inside the red blood cell, which has a more normal ratio of pH on the inside? It turns out that the drug, the orange drug, the singly charged drug, will stay on the inside of the inner leaflet of the cell, which is really good because that is the side of the membrane that we want the drug to be on. It's the side that is inside the cell. And the reason why will be shown by this group. They calculated two ways, and so they calculated a drug partitioning. This is like what I showed you, and I just want to show you they do this really good example of how the drug partitioning could work if you have two pKa's rather than one. And that's the way that 5 of the antimalarials actually work. They're small molecules. They have 2 pKa's on them. The pKa values, of course, determine whether it's charged or not. And the standard way to talk about whether it can cross the membrane is to calculate the ratios using Henderson-Hasselbalch and to say, an example like what's shown right here. You have the solvent outside the liposome. You have the core inside the liposome. Those are at different pHs. And you can have some form of ion trapping like we had at the beginning of this talk, where you can have, if you can have the pH such that the drug becomes unionized, non-ionized, it will then pass through the lipid bilayer, get inside, and adopt the pH, the charged form that goes with the pH on the inside of the liposome. This is basically a model. This looks like the ion trapping that we just talked about. It just adds on another pKa because we have another group that can be charged or not. So a simple model is just like that. It's just like what we mentioned. The group actually has a more complex model. They started with a more advanced model because they realized that actually the singly charged drug, if it looks like this, okay, so if it looks like it has a charged group on the very end of the molecule, on a primary amine that is sticking out from the molecule, then there's going to be, if it has anything of that form can actually also dissolve in the membrane. It's going to be hard for it to cross the membrane because it's going to be hard for that single charge to get across the membrane, but it will be able to dissolve in the membrane. And so you should account for that being able to cross and partition across the liposome as well. And they're showing that. They're adding that reaction into their modeling. This was actually a computer study, so I just want to say it goes along with experimental data, but it is not it's not proven yet. Nonetheless, I want you to see, oh, this is how a drug like this that is the right size can actually dissolve. If it's about the same size as a membrane lipid, it can actually dissolve in the membrane, and it can have its charged NH3 group on the outside of the membrane, just like the polar head group of a phospholipid. And so that's actually, they use this, they actually developed this model to account for the fact that we could see some of this happening experimentally. Notice at the bottom you have the dimensions in angstroms of how wide the membrane is. And notice that the drug actually can fit very well into one leaflet of the membrane. This physical compatibility is one of the things that makes this work. It may be one of the things that allows antimalarials to get across a membrane. So what they did is they show right here what the different forms of the drug will be at different pHs with the pKa. This is actually just an application of the Henderson-Hasselbalch equation to each of these pKa groups. Each group has the pKa, and they've basically done a plot where they have pH across on the x-axis and just the abundance, the percentage of it that's in that form. And so you'll notice that at low pH you have the doubly protonated form, which is going to be the green form. Because these are both basic, that's going to be charged twice, and that's not going to have any chance of getting across the membrane. At the other side of the graph, in the gray part, and when you get up to pH 10 or 12, then you actually have both of the pKa's, you're above both of the pKa's, you've deprotonated both of them, and the molecules are therefore neutral. They show that as the gray part of the graph. In between, you have the singly charged, which is shown as red. The singly ionized form is going to have a single charge on it, and the good news is these single charges, the pKa's that are higher, are the primary amines that are on the end of the molecule. Because of this, the red form, the singly charged form, can actually dissolve in a membrane. These three antimalarials all act the same. Look at the graphs, and they all have similar ranges. They are at neutral pHs, they are singly charged. And that's because their pKa's are similar to each other. You look up and down at those two pKa's, they're both around 10, 9 to 10, or 3 to 4 for the two different pKa groups. That means their pKa2 values are low. These are singly charged at neutral pH. However, there are two, oh, by the way, I should read them out. Quinine, primaquine, and tafenoquine are the three drugs that are like this. But there are three other drugs, and you can see these are similar sizes, and they also have two pKa's that behave the same way. But the pKa's have different structures to them, and they have different values, and so their pH behavior is different. These drugs at neutral pH are green, not red. Green means that you have two charges on them, and that means that much less of a chance of getting across the membrane at neutral pH. So they have quinacrine and chloroquine that are shown in this way. These are going to have different charges at neutral pH, and they will behave differently because they will have two charges on them at pH 7 instead of just one. So this does fit with how things look. These are arranged so that they have the doubly ionized drugs on the right side. They have all 5 of the antimalarials. The first 3 are singly ionized at neutral pH. They're the red ones. The second 2 are doubly ionized, which were green on the previous graph. And they find that it's hard to get the doubly ionized drugs to be encapsulated in the liposomes, because even at low pH, they are far too charged all of the molecules have 2 charges on them instead of some of the molecules having 1. And so these doubly ionized drugs at pH 4, they don't go into the liposomes. If you look at this graph right here, here's the pH 4 graph, and you can see that the 2 bars on the right, the doubly ionized drugs, are not able to get into the liposomes. The singly ionized drugs tend to be more compatible. They tend to have the higher bars. There's some complexity here, but the 3 bars on the left will tend to be more encapsulated in the liposomes than the 2 bars on the right. That's what this has shown. And the effect goes away when you raise the pH and make the liposomes at high pH, because there, all of the drugs are more deprotonated, they're more non-ionized, and they're more able to get into the liposomes and even dissolve in the liposome membrane. So this is what this makes sense. What they were observing really is that you actually have pretty good loading at pH 4 for at least 2 of the singly charged ones. You have 2 of the bars on the left that are really high on this part of the graph. And they're also high at pH 6.0. So why are the singly ionized drugs compatible with the membrane at all? Well, that's why they developed their theory that they do this, that they are able to cross the membrane because they are able to fit into the membrane with their charged part outside the membrane. That's what the important part is about how structure and function fit together here. They also noticed something interesting. When they can make the liposomes with different phospholipids. And the 2 that are important to me are phosphatidylcholine, which is sort of the normal default one that they use for most experiments. It's a very common phospholipid in human membranes. And so you can make the membranes that way, but they also tried different phospholipids, and so they tried some with phosphatidylserine mixed in. That's shown as PC:PS. So the PC is phosphatidylcholine only, PCPS is with phosphatidylserine. And the one thing I'll have you notice is that for all 5 of the drugs, you end up with higher bars for the PCPS line, the second one over, than for the PC line by itself. When you add in the PS, you get higher bars in this second area over here. I can show you it right there. So what is it about the PTD the phosphatidylserine, that is compatible with the ionized drugs? Well, they use this as more proof of their model, because if you compare phosphatidylcholine to phosphatidylserine, you'll see that the difference is in the head groups. The difference is not in the lipid chains shown at the bottom. The difference is in the head groups shown at the top. Phosphatidylcholine, the choline group, has a single positive charge on it that offsets the negative charge of the phosphate. Phosphatidylserine also has a positive charge on it, but it has a second negative charge. So it ends up that phosphatidylserine has an overall negatively charged head group, whereas phosphatidylcholine's is overall neutral. Think about how that goes together with their idea that the drug dissolves in the membrane and the NH group, the pKa1 group, is actually sitting outside the membrane like a head group. It would be right next to the other head groups. So if you had phosphatidylserine in there, it would have a positive-negative electrostatic stabilization. Positive and negative charges attract. Phosphatidylserine and the charged pKa1 group of the drugs attract. The other phospholipids are more neutral, and so they're saying that this would explain why the drugs are able to get in, and they show it, you can see it on their graph sort of this way. You would have negative serines carboxyl groups as the head group wherever you have phosphatidylserine. So that would help their vesicles, their liposomes, which have phosphatidylserine just mixed in randomly. But the really interesting thing is red blood cell membranes also have a lot of phosphatidylserine, but red blood cells organize all of their phosphatidylserine on the inner leaflet. If the drugs really work this way, that means that we can expect them to be enriched on the inner leaflet of red blood cell membranes. So when they get into the red blood cell membrane, they work better because they are able to be stabilized by the extra negative charges that interact with the positive charges on the singly charged drug that has to be there a lot at neutral pH. This could explain how these antimalarials are able to get into the cell even though they are singly charged at neutral pH or doubly charged, because there's enough of them can dissolve in the membrane and be stabilized on the inside of the membrane by the high concentrations of phosphatidylserine. By the way, on these figures where you have like a lot of text on the bottom of it, I don't intend for you necessarily to look at that text. I want you to look at the words that I've written and hear my explanations that I'm giving you. The text is for more information for those who might want to get into the technical actual literature. I invite you to see that you can probably take it apart with time, but for this class, I am more interested in you understanding the concepts that I'm teaching through the figures. I'm not really interested in you digging down into the captions. And so with that, I'm going to give you another couple of examples of interesting interactions of pH with membranes and drugs. Okay? There are a class of drugs that can actually dissolve in the membranes and disrupt the membranes, and these can be pH dependent. For example, here is a paper where they screened for lipophilic molecules that can actually dissolve in a membrane at a certain pH and destroy the membrane by somehow interfering with it. Bacterial membranes, by the way, are weaker than human cell membranes, and so this could be a way to have an antibiotic. You can have a molecule that will destructively interact with particular membranes under particular pH conditions when they are ionized in the right way. Here's some text from it. They miniaturized a high throughput screen to identify compounds that disrupt pH homeostasis. They basically looked for disruptive compounds that they would mix them together with their organism. And if you look at the organism, it's Mycobacterium tuberculosis. They basically screened 90,000 small molecules. Out of that, they found about 500 that actually resulted in the bacteria having pH that was too low. That's an indication that these are making the membrane disrupted. They're making it leak. So out of those 500, they selected 5, and they showed that they actually would work against bacteria and not work against, not disrupt human membranes, or not even disrupt. Even better than that, they found that they disrupted non-replicating bacteria because those are the ones that would be more stressed by the pH disruption. I just want you to see what these compounds look like. These compounds look like the antimalarial drugs. They are about the same size, about the same molecular weight, about the same lipophilicity. Look around at the nitrogens, look around at the different groups they have. These are rather non-polar molecules. They're lipophilic. And they all have ionizable groups that are nitrogen bases, just like those. Maybe those fit into the membrane like the drugs fit into the membrane. And this also applies to peptides. Some animals actually make peptides that go into bacterial membranes and they dissolve into them and they disrupt them, just like the small molecules do. Clearly, these peptides are much larger than the drugs we're talking about, but they are usually half nonpolar, half polar. So their nonpolar parts dissolve in the membrane and their polar parts disrupt the membrane. Here are some actual sequences and just examples. I just give you to these as examples. These usually are short sequences. They're made by organisms such as the African clawed frog. The honeybee makes one. The, I can't even say that, sauvage leaf frog. And even humans, we make a sequence that will actually, a sequence of proteins that will actually disrupt bacterial membranes. And so that's kind of cool. And what I get this from is these antimicrobial peptides are actually very effective, especially against neglected tropical diseases. Here is a paper that talks about how they are effective against African trypanosomes, leishmaniosis, and Chagas disease, trachoma, and even leprosy. So these do a good job on taking care of bacteria, and it's possible that one of these will come in useful to you in your medical practice someday. And so it's kind of helpful for you to know that these kind of peptides, yes, they are bigger than the nonpolar drugs that we were talking about, but they work by disrupting the membranes, and we're talking about things that are lipophilic, dissolving in membranes, disrupting their structure and function, basically punching holes in the membrane so that then stuff leaks inside and out and the bacterium is lysed. And that brings me to my last topic. I want to bring you to, if we understand this well enough, we can actually design things that will work in a certain way. And my field is protein design, so I want to show you what a protein designer can do to make a protein that will disrupt the membrane only at a certain pH. So this is using the pKa of that protein, of a group on that protein, to make the protein work at one pH and not work at another pH. So the design has been, let's make some proteins that are short sequences like the ones I just showed you, short sequences like these. Let's design some proteins that will reliably only disrupt membranes at acidic pH values. So we put it in a situation where in an acidic environment, it will disrupt membranes, but at a neutral pH, it will not. And this could be good for keeping the patient, who's mostly at a neutral pH, keeping the patient safe from these membrane disruptions. Right here, it's a general strategy to design pH-responsive protein conformational changes by precisely pre-organizing histine residues in buried hydrogen bond networks. Two words to notice: histine residues and hydrogen bonds. We're talking about something having to do with pKa. By the way, the word de novo design means that they didn't start from another example. They started from a computer. So a computer designed these from the bottom up. I think that's pretty amazing. This is what the proteins look like. So these are homotrimers and homodimers. It means that they are alpha helices that are 3 chains or 2 chains that come together, and above pH 6.5 at neutral pH, they are stable. And so they just basically sit around, and they flow through solution like normal proteins. But when the pH gets low, they suddenly fall apart and they reveal their insides. Their insides are part of the antimicrobial activity, the anti-membrane activity of these. What makes this happen is they have a histidine. See this histidine 45 right here? That is a histidine that is right in the middle of the protein. When that histidine is at neutral pH, it is deprotonated and is able to participate as a hydrogen bond acceptor. But when that gets protonated, it can no longer be an acceptor. It would have to be a donor. And that makes the whole bond break and the whole protein fall apart, which results in membrane disruption. Realize that the key to this is one tiny proton going on to one histidine residue, and everything else falls apart. They compare 2 examples here. They have this Pro2 protein, which has the histidine in it that they've designed. They have a Pro2 no-his version, which looks exactly like the histidine version, but instead it has an asparagine. Asparagine does not, it forms similar hydrogen bonds, but it does not ionize. It's not got a pKa that is anywhere near the histidine pKa of 6.0. And so these are really good examples. We can see that the histidine one should be pH sensitive, whereas the other one should not have the pH sensitive effect. Remember this. Remember that histidine has a side chain with a pKa value for the side chain, the R group, around 6.0. That means when you get below 6.0, the histidine will absorb the proton, become positively charged, and because this nitrogen was previously a hydrogen bond acceptor, once you put a hydrogen on it, it can no longer be a hydrogen bond acceptor. And that one hydrogen bond is broken. And this is their picture of what it looks like. I just want you to see the histidine right here. Here it is a nice little hydrogen bond acceptor on the right, and on the left side is forming a hydrogen bond to a hydrogen bond donor. At low pH, it becomes protonated, and both of those, you can't have you can't have a donor bonding a donor. You have to have a donor bonding an acceptor. They clash, and the protein falls apart. So when the histidines are protonated inside the protein core, the core of the peptides fall apart, the helices fall apart, and then they expose the hydrophobic residues that will go dissolve in the membranes and punch holes in the membranes. They will kill the particular cell that has that membrane in it, and they will kill nearby bacteria as well. This is how they prove that this is going on. Each graph goes with the protein that it's next to. And notice that pH is along the x-axis. At a neutral pH around 7, you see the black line is high, which is the trimer. They measured how much trimer and how much monomer they have. When you get to around pH 6, the trimer falls apart. That's exactly where the histidine would be protonated. And then you have the monomer line, the gray line, increase. For the no-hist version, the black line stays pretty much the same. The trimer concentration just stays there. It doesn't care that you are reducing the pH. It's stable because it doesn't have the histidine to be protonated. Here's how they show, and I just have to show you some actual protein folding data. Right here, I just want you to notice this is where they test the stability by adding a molecule called guanidinium chloride that will actually unfold the protein, and they want to see how it unfolds at different pHs. Red pHs are lower than the black pHs in this case. Notice that in this graph, let's start with the lower graph. The no histidine version has all the same, all the pHs line up. It falls apart at about 2 molar guanidine. And by the way, the higher the line, the more unstructured, the more structure you have lost here. But notice that you see all the different colors not lining up anymore for the Pro2 protein on top. If you look up here, you see that the red line and the yellow line actually are, they don't show the sharp transition, which is a sign of a stable protein unfolding. In fact, it looks like they've already been unfolded. So this shows that they don't have much structure. The important thing is the red line doesn't have a sharp, steep transition. The steeper the line, the more stable the structure. The red line is not steep at all. In fact, it looks like just a flat slope just going up. It's hard to see where this is falling apart because it has already fallen apart at pH 4.3. This is one last thing where they measured, they basically measured how these designed proteins will actually disrupt lipid membranes in vitro, which means in a test tube or in an experiment, and in actual cells after they're endocytosed into mammalian cells. They measure this with a dequenching signal. Basically, this is how leaky is the membrane. The more leaky the membrane, the higher the signal. And so you see right here, the red lines are lower pH with more protons around. The more protons you have around, the higher that red line. And the red lines show that at lower pHs, the membranes are suddenly leaking all over the place. The more leaky membranes, this goes along with their theory that the protein is doing what they designed it to do. Notice also that they compared using the Pro2 protein at pH 5.0 to the Pro2 no-hist protein. The Pro2 no-hist protein doesn't leak at all. But if you add the Pro2 protein and if you have the pH down at 5.0, all of a sudden you have a leaky membrane because the peptide is punching holes in it. Remember that we had this story about endocytosis, and I said this would be important. Remember that you have cells form some vesicles where they take things in from outside, and then they acidify them. They bring the pH down to about pH 5.5 or pH 5. Well, if you have this protein working like it should, the endosome would gather it in, the pH would lower, and then the protein would punch holes in the endosome. And they tested to see if it would work in the complex system of a mammalian cell. And they found out that it did. Finally, this is, again, we have complex captions. I don't want you to know the details. I want you to know that this shows pH-induced disruption in endosomes. For example, right here, this first is taking, it's using cryo-electron microscopy. It's basically using a form of microscopy to, and it's using gold nanoparticles that you can see. It's putting the gold nanoparticles on the proteins, and they see that the proteins go right up next to the membranes. The proteins are located where they should be, right at the membranes. And then when they add the proteins to the membranes, and then they do a pH 5.0 solution, all of a sudden you see this. Look at the membranes are disrupted. At pH 8.0, or with the Pro2 protein, you see nice circular membranes. But on the left, in the two things where you have pH 5.0 and the Pro2 protein, you literally see the membranes disrupted. That's what I want you to see. I want you to see these membranes are spilling out. It looks like the protein is working. Now, this is with artificial membranes and liposomes. What happens with real cells? This is a little bit harder to see. I don't want you to look at this too closely, but I just want you to see they show evidence right here that it's actually working. It looks like you put this with mammalian cells and it will disrupt the endosomes. In this case, it disrupts a type of endosome called the lysosomes. This is where they prove it. This is biology. I'm a chemist, and so I don't really care that much about this, but I want you to know they have proof, and they can see this thing working in the liposomes in real mammalian cells, which is pretty nice because you designed this from the ground up. You designed this with a protein. So that's what their conclusion is. They actually want to use this, and this is, again, for your own information. It's a detail that doesn't really apply, but who knows? Maybe someday you could use a pharmaceutical that's developed on this. Because their idea is you could attach like an antibody, a toxic antibody or a toxic protein to the trimer, feed it to an animal, and feed it maybe even to a tumor. And maybe it'd be endocytosed, the pH lowers, and then all the endocytosis destroys the endosome, and the pharmaceutical is delivered to the entire cell. So if it was a beneficial pharmaceutical, I used an example of a toxic pharmaceutical, but what if it was a beneficial pharmaceutical? This could be a way to deliver the pharmaceutical to the inside of the cell, to get it across the membrane, which is a very hard thing to do. This is their actual words about it, and the only way we really have to do that right now is to use things like other kinds of protein designs or viruses. And this is actually related to gene therapy. So I'm going to leave all this here, but you don't have to, I'm not even going to read it. I just want you to know they see a use for this, and who knows what will happen in the future. If this comes about in the future, if you know how membranes work, if you know how pH and pKa can help disrupt membrane function and can help things get across membranes, depending on the situation, then you will know how to deal with this. That's my version of this. I'm sure that Dr. Bond will have a lot of clinical applications that he can mention about this. This is my biochemical application. So thank you for listening, and I will go and send this off to you now.